Hello children, today let us read about Biology Standard 9, Digestive System, Unit 3. In the last class we read about the alimentary canal, the digestive system, the dentition and the structure of teeth. Today we will learn about the gastric juice, bile, the pancreatic juice, intestinal juice, assimilation of food, liver and few experiments on digestion. Means that is the digestive system consists of the digestive glands. Now the food after entering into the stomach. Stomach is an elastic bag located in the diaphragm and the food reaches the stomach with the help of peristalsis. Now the gastric juice from the stomach is secreted from the inner lining of the stomach. This gastric juice is a colorless, highly acidic liquid containing water, salts, hydrochloric acid and contains an enzyme called as pepsin. This hydrochloric acid serves two functions. It kills any germs which may have entered along with the food. It activates the pepsin to act on the proteins. Now let's see this reaction. The pepsinogen, which is inactive in the presence of HCl, converts into pepsin, which is active. The proteins with the enzyme pepsin converts into peptides. The prorenin, inactive state, changes to active state into renin, and the milk protein soluble form changes into insoluble form in the presence of the enzyme renin to form paracase. Now this food stays in the stomach for about 3 hours and attains a pulp-like form called as chyme. Now the pylorus opens to allow the chyme to move into the intestine little by little. Now this is a diagram of the anterior view. You can see the gallbladder here, the liver, the stomach, the pancreas, a leaf-like structure, the duodenum and small intestine with large intestine. The small intestine serves both for digestion and absorption, which receives two digestive juices. One is a bile and the other is a pancreatic juice in the duodenum, and its own walls secrete the intestinal juice in the ileum. Now what is bile? Bile is a yellow color, green, watery fluid produced in the liver, which is transported through the hepatic duct. The color of the bile is due to the pigment called bilirubin and bilirubin produced by the breakdown of the dead and worn out tissue, RBC. The bile salts neutralizes the acid content of the food, makes it alkaline to enable the pancreatic and the intestinal enzymes to act. It reduces the surface tension of fats, breaks them into tiny droplets for the action of enzymes, which is called as emulsification. The fat with the help of bile changes into emulsified fat, and the acidic chyme in the presence of sodium bicarbonate changes to alkaline chyme. Then what is pancreatic juice? This pancreatic juice is produced in a whitish gland called as pancreas, which is located near the stomach, which is a leaf-like structure. The pancreatic duct opens into the duodenum by an aperture called as bile duct or the common tube called as the bile duct. The pancreatic juice contains 3 types of enzymes. Amylopsin, that is pancreatic amylase, which digests the leftover starch into maltose. The trypsin, which acts on the remaining proteins and polypeptides to produce smaller peptides and amino acids. Steepsin, which acts on emulsified fat to split them into fatty acids and glycerol. This is a diagram of the pancreas, the pancreatic duct, which is a leaf-like structure, the liver, small intestine and the bile duct. Now this is a reaction for overall pancreatic enzymes, where the trypsin changes in the presence of the enzymes enterokinase to active trypsin, proteins and peptides in the presence of trypsin to form smaller peptides and amino acids. The leftover starch converts into maltose in the presence of the amylase and emulsified fat in the presence of steepsin to fatty acids and glycerol. Next, what is assimilation of food and the structure of liver? Assimilation of food means the conversion of the absorbed digested food into the body material, where we call it as assimilation. Now, conversion of the excess glucose into insoluble glycogen is called as glycogenesis. The reconversion of glycogen into glucose is glycogenolysis. Excess of amino acids are broken down in the liver by a process called as deamination. Now, what is liver? Liver is the largest gland of the body, weighing about 1500 grams on an average. It is reddish brown organ located in the upper right side of the abdomen, just below the diaphragm. What it does? It controls blood sugar level. It helps with the synthesis of RBCs, stores iron, copper, vitamins A and D. It helps for detoxification. It produces heparin. This is a diagram of the liver, where there are two lobes, the right lobe and the left lobe, the coronary ligament, the gallbladder is here, and the falciform ligament. Now, intestinal juice contains erepsin to convert the peptides into amino acids, maltase to digest maltose into glucose, lactase to digest lactose into glucose and galactose, invertase to split sucrose into glucose and fructose, lipase to digest fats and fatty acids, and glycerol. This is a reaction which takes place for the intestinal juices. All the changes are given as summarized. The peptides changes to form the amino acids in the presence of peptidase, maltose to glucose, maltase, sucrose forms glucose and fructose in the presence of sucrase, the lactose, which is otherwise called as the milk sugar, to glucose and galactose, and emulsified fat to fatty acids and glycerol in the presence of lipase. Now, let us learn some experiment on digestion. One experiment we can do, the action of saliva on starch. So, we can prepare a suspension of some starch by heating 1 gram of it in 100 ml of water in a beaker. We will put the suspension in 2 test tubes and we will test the sample 1 of the test tube for starch by adding iodine solution and the other sample from the other test tube for sugar. Let's take little starch suspension in a third test tube and add to it an equal amount of saliva, which we may collect by chewing some wax or chewing gum. We will hold the test tube in our hand for 15 minutes to keep it warm, or we can heat containing in a water beaker of about 38 degrees centigrade, and we will perform the sugar and starch test on the samples taken. It will no longer give us a positive test, but will show the presence of sugar. Now, here let us learn to the presence of starch, glucose or sucrose, proteins and fats if given in a simple or in a sample of food. Now, when we learn for the test for carbohydrates and starch, let's take 2 to 3 ml of sample solution in a test tube and add iodine solution, and what will we get the observation? The solution turns blue-black, which proves that the given sample contains starch. Same way we can go for glucose by taking 2 to 3 ml of sample solution in a test tube and we'll add 3 ml of Benedict solution and heat it. The blue solution after heating changes to orange and then to brick red precipitate is formed, which proves that the sample contains glucose. When we test for a non-reducing sugar sucrose, let's take 5 ml of given sample solution in test tube add 1 ml of sodium hydroxide and 1 ml of cobalt nitrate. The color of the solution changes to violet color. This color change shows the presence of sugar. Test for protein. We can take 5 ml of sample solution by adding 3 ml of sodium hydroxide and 2 drops of 1% copper sulfate solution. The solution turns into violet color or mauve color, which shows the presence of protein. Test for fat. A little bit of food sample like butter is taken and rubbed on a white paper. The paper turns greasy and translucent, which confirms the presence of fats. So in this way we can learn some of the experiments for presence of water, presence of minerals can be done. With this we complete the chapter of digestive system, and in the next video we will go for the next system. Thank you.